All right, good morning, everybody. Okay, so let's get started. Today is Tuesday, right? Thursday. Last week, and we started talking about the lipoproteins, right? And we finished the two triglyceride-rich lipoproteins, right? I have a couple questions later. We will, do the kind of practice, right? So chylomicron and VLDL, right? And we also talk about the metabolism of these two triglyceride-rich lipoproteins. And we all know that lipoprotein, the main function of lipoprotein is to transport lipids, right, between tissues. And we have a different type of lipoprotein. That means each specific lipoprotein, they have specific functions in terms of type of lipid as carries from tissue, one tissue from other tissues, and the other important thing that each lipoprotein carry lipids from specific tissue destination, right? For example, chylomicron is a lipoprotein that is synthesized in the small intestine, and the main function of chylomicron is to transport triglyceride from small intestine to which tissues mainly? Adipose tissue for storage, right? So what do you think, in what physiological conditions chylomicron should be synthesized a lot? Yeah, in the fat condition, right? When we eat a meal and we absorb a lot of lipids, triglyceride, we need to put the lipids that we absorbed from diet, into the adipose tissue for storage. And VLDL, which tissues synthesize VLDL? Liver, right? Liver is the main tissue that synthesizes VLDL. And also I
mentioned depend on, again, the physiological condition. The liver can synthesize VLDL in both fat and fasting condition, right? Not like chylomicron mainly synthesized in the fat condition. So the fat condition, VLDL is synthesized in the liver. The main purpose of VLDL is to transport triglyceride, right, from liver to adipose tissue for storage under the fat condition. And the triglyceride that being packed into VLDL mainly from, I'm talking about the fatty acids that are in the triglyceride, mainly from lipogenesis, right? Under the fat condition. But during the fasting condition, VLDL can also be synthesized in the liver. And the purpose of VLDL under fasting condition is to transport triglyceride from liver to which tissue? Muscle, yes, thank you. So muscle. So muscle picks up fatty acids from VLDL and utilize for energy, right? So again, you needed to think, understand under the different physiological condition, which tissue synthesize which lipoproteins, what is the purpose, right? The tissue synthesize lipoprotein, and which is the destination of that tissue that lipids being transported to. Now let's. Have you guys get a chance to watch the video? Yeah. Okay. So I think it's pretty helpful, right? And help you understand the composition of different lipoproteins. Now let's talk about HDL. So both HDL and LDL are the cholesterol-rich
lipoproteins, right? That means that most of the core lipids are the cholesterol ester actually, right? So obviously, I mean, this two lipoprotein HDL and LDL basically deal with the cholesterol metabolism, right? So HDL core lipids. What do you expect the core? What types of lipids are mostly? And HDL, cholesterol, right? Cholesterol esters, and also some triglycerides. Apoproteins that are present on the surface of HDL are ApoA1 and ApoA2. And what are the where HDL synthesized? Liver and intestine, right? And the next question is, what are the functions of HDL? We know just in general deal with cholesterol, right? Cholesterol metabolism. But specifically, what HDL does compared to LDL, right? They both lipoprotein does the opposite function. So HDL, what HDL does is to transport cholesterol from peripheral tissue back to the liver for disposal, right? And LDL basically is to dispose the cholesterol into the peripheral tissue, right? So we also talk about the concept that is very important. It's called reverse cholesterol transport, right? So reverse cholesterol transport means that the HDL transports cholesterol from peripheral tissue back to the liver, right? And this reverse cholesterol transport process is accomplished by so-called HDL cycle, right? So we have, in our body, we have two different types of HDL, called alpha HDL and pre-beta HDL. And these two HDL basically can interconverted, right? And the main difference between these two type of HDL is the core lipids, right? Now let's talk about the HDL cycle. So again, this cycle is accomplished by the conversion between alpha HDL and beta pre-beta HDL. So the function of these two different type of HDL is different. So alpha HDL basically is function. Its function is to give the cholesterol to the liver. So alpha HDL has a core that has a lipid core, right? Let me just explain the the composition of each HDL that can help you better understand, right? So, for example, pre-beta HDL is considered the empty dump chunk, right? You know, the purpose of this, we can consider HDL as a dump chunk, right? So the lipids, particularly cholesterol esters, we consider as the trash or whatever, right? They need to be, moved away and disposed in the liver from the peripheral tissue to the liver. So beta HDL usually has no or little core lipids. So it's like empty, again, the dump chunk, right? But it still has, some of course, the phospholipids, right, the surface lipids, and also proteins, ApoA. But if you compare to the alpha HDL basically is considered a full dump truck. That means you uploaded all the lipids, right, already, in the HDL. So, and this alpha HDL is filled of the cholesterol in the core, right? So again, the main difference between these two HDL is its content, right? The lipid content. The beta HDL, beta is empty, right? Empty, the truck, and the alpha HDL is full of the lipids, right? And this diagram give you like overview how reverse cholesterol transport happen, right? Again, the peripheral tissue basically like artery wall. They have a different type of cells, endothelial cells and macrophages, right, on the artery wall. And the lipids, particularly free cholesterol and phospholipids, PL, can be uploaded, right, into the beta HDL. Beta HDL, again, basically is considered empty, right, and dump truck. And once you get enough, once beta HDL gets enough lipids, free cholesterol of phospholipids from peripheral tissue. Here we talking about the artery wall, right? And they become alpha HDL. So from small to the large, depending on how much lipids were loaded, right? So and eventually the alpha HDL and carry all the lipids from peripheral tissue, from artery wall to the liver, and dump the cholesterol esters into the liver, right, and for disposal. And alpha DHL it's losing the cholesterol, right, losing lipids, then it become empty HDL, beta HDL again. And that beta HDL can go back to the peripheral tissue and load in more lipids and become again alpha HDL, right, and carry the lipids to dispose the lipids in the liver, right? So this process called is the reverse cholesterol transport, right? But there are also several things happening in the plasma. For example, initially the cholesterol is loaded loaded in the beta HDL is a free cholesterol, right? So free cholesterol, usually located on the surface, right? The lipoprotein, beta HDL. But eventually, this free cholesterol need to be reacidified to cholesterol esters and then enter the inside of the lipoprotein. That means in the core, enter the core area. All right. There are actually two important enzymes that are present in the plasma that catalyze the reacidification of free cholesterol to cholesterol ester. And also there are other things happening, lipids exchange, I mentioned earlier, right, between the different lipoproteins. So all these things work together that makes the lipids composition in the HDL changes. Both amount of lipids and also composition of lipids are changed, during the transporting or traveling from peripheral tissue to the liver, right? Any questions? Right? And the purpose of this process is just to dispose unnecessary cholesterol, right, from peripheral tissue. Here we talking about artery wall, so that can prevent the accumulation of cholesterol on the artery wall, right, to prevent the development of atherosclerosis. So that's why HDL we consider good cholesterol, right? It's very important. We need to have a higher cholesterol level in the circulation. If we have, for some reason, we have a lower, or some people have a lower HDL, then most likely, have a high risk for the development of atherosclerosis. Here is a couple of enzymes and proteins that are very important, right? Regulates this reverse cholesterol transport process. First one is ABCA1. ABCA1 is ATP binding cassette transport A1, right? This is a protein that located on the cell membrane of peripheral tissue, right? If you go back to the last slide, see here, the peripheral cells, the surface, right, has a ABC1. And the function of this protein, is to help transport of lipids, free cholesterol or phospholipids, right? Help transport these lipids from peripheral cells to the beta HDL, right? So if we don't have, or we have somehow the lower levels of ABC1, what kind of problem that we will have? If we don't have enough ABC1, some reason in our artery wall, cell surface. Yes? Yeah, cholesterol cannot be, right, an output or an export, right, from the peripheral cell to the beta HDL. So then we will have, what do you expect, like levels of beta HDL or alpha HDL in the circulation if we don't have enough ABC1? More beta and less alpha, right? So if we measured, using kind of a technique to measure the beta and alpha HDL level in the circulation, if the patient had less ABC1, then the patient should have a higher beta HDL and lower alpha HDL. So, okay. And the second receptor is also important, is SRB1. SRB1 is a scavenger receptor B1, right? It is also, it is located on the liver cells, but also other peripheral cells as well, right? And this receptor basically is functioning in facilitating the uptake of cholesterol from HDL. Sorry, I think somehow I missing M, I guess. So the facility uptake of cholesterol, right, into the cell. All right. So the third one is the LCAT. LCAT is a lecithin cholesterol isotransferase, and this is enzyme catalyze the aspiration cholesterol to cholesterol esters. And this enzyme actually present in the circulation in plasma. And we also have a similar enzyme catalyze the same reaction, the acidification of cholesterol to cholesterol ester, but the location is different from this, from LCAT, which is ACAT. So ACAT is the same, have the same function, right, as the LCAT, but it localized in the inside of cells, right? So again, this enzyme also catalyzes the acidification, right? Acidification, free cholesterol to cholesterol ester, but again, the different location. And the last one is the CEPT. CEPT is cholesterol transfer protein present in the plasma. And this protein, the main function of this protein is to facilitate exchange of lipids between the different lipoproteins, right? So try to remember all these different receptors and enzymes, proteins, right? And get to know, where this protein enzyme is located and what are their functions, right? That can help you to understand the whole process of a reverse cholesterol transport or HDL cycle. So this slide will just give you more detail, but similar. We'll just get into the more steps, right? During the reversal transport process. Again, this is the liver and the peripheral tissue, the arteries, and between this tissue, all these things actually happening in the plasma, right, the circulation. So first of all, let's start with the beta HDL, right? It is empty tract, right? And try to get the lipids from the peripheral tissue, and on the peripheral tissue surface there is ABC1, right? That control the transport of lipids from peripheral cell to beta HDL, right? And the two lipids that are transferred from peripheral cell to the HDL are the free cholesterol and phospholipids. So once the beta HDL gets all the lipids from peripheral cells, then become the so-called discoidal HDL and then released into the circulation, right? And in the circulation of plasma, the free cholesterol that initially get from the peripheral cells and gets to the acidification to cholesterol ester, and this is the enzyme LCAT, right, present in the plasma that catalyzes acidification of free cholesterol to cholesterol ester. So once the cholesterol ester is formed, the cholesterol ester will enter the core, right, from the surface to the core, and eventually the beta HDL becomes alpha HDL once it is filled with the lipids, right? And also something happening between, the lipid exchange between the other lipoproteins, HDL and other lipoproteins. So initially, alpha HDL mostly or only has the cholesterol ester, right? And after exchange lipids with a VLDL and IDL, LDL, the alpha HDL gets more and more triglyceride and gives some cholesterol esters to VLDL or other two lipoproteins. So eventually, the alpha HDL become still cholesterol rich, but it also has some triglyceride, right? This triglyceride is actually from the other lipoproteins. Does that make sense? Right? So about 30% of initial cholesterol ester that was picked up from the peripheral tissue goes to VLDL, IDL, or LDL, right? So that makes VLDL remnant, IDL become eventually LDL, because LDL gets more cholesterol esters, right, from the HDL. So eventually the alpha HDL that gives all the lipids, cholesterol esters, and triglyceride to the liver, right? And liver disposes the cholesterol in the several forms. For example, can synthesize bile acid from cholesterol and also steroid hormones, right? And alpha HDL, once it's uploaded its lipids to the liver, then it becomes back to the beta HDL, right, and goes back to the peripheral tissue and do the similar same cycles. Any questions? Right. Okay, so just the more details. So HDL cycles basically is a conversion, right, between alpha HDL and beta HDL. So basically these two types of HDL has different composition, particularly the lipids, core lipids, right? So what happened to alpha HDL when it becomes beta HDL? So beta HDL is empty truck, right? And alpha HDL filled with trash, like cholesterol. And so several things, right, happened to the alpha HDL. First is a reduction of core lipids, right? Core volume, basically loss of cholesterol. And second, the dissociation of the ApoA1. It's also lost ApoA1, right? And then the third is the internalization of a cholesterol ester from alpha HDL. And lastly, the triglyceride that is transferred from VLDL or IDL, right? That can be hydrolyzed in the alpha HDL by hepatic lipase, right? Because the alpha HDL eventually picked up by liver, right? And this all cholesterol esters and triglyceride in the alpha HDL pick up, by liver and hydrolyze in the liver. So then the hepatic lipase should be the important hydrolysis enzyme, right? Does that make sense, right? So this is actually happening in the liver. And conversion from beta HDL to alpha HDL, this process occurs in the peripheral tissue and also in the plasma, right? So what happens to beta HDL when it converted to alpha HDL? First, you pick up the lipids, right, from peripheral tissue. So in general, the beta HDL should be abundant in the peripheral tissue. So pick up lipids, free cholesterol and phospholipids, right? And then the ABC1 is important transporters, right, that control the flux, so-called efflux of the free cholesterol from peripheral tissue to alpha HDL. To beta HDL. And once beta HDL receive enough free cholesterol and phospholipids, and then converted to alpha HDL, right? Of course, have several things, at least two important things happening in the plasma during this conversion. One is the reacidification, right, by LCAT. And the other thing, the second important thing, is the lipid exchange that characterized by CETP, right, protein in the plasma. Okay, any questions? All right. There's something I just talked, something happened within the plasma, two things, right? During the conversion from beta HDL to alpha HDL. One, again, is the acidification of free cholesterol to cholesterol esters, right, by LCAT. And once it's acidified, cholesterol esters move into the core from the surface. And the second important thing is the exchange of cholesterol ester, triglyceride, between alpha HDL and the VLDL, IDL, LDL, right? And eventually the alpha DL filled with the core lipids, particularly cholesterol ester and triglyceride, and picked up by liver, right? And scavenger receptor is important receptor that control the uptake of the alpha HDL into the liver cells, right? And eventually in the liver, the cholesterol esters being released from alpha HDL and be disposed in the form of a bile acid, and also cholesterol can be used for steroid hormone synthesis. And triglyceride in the alpha HDL can also be hydrolyzed in the liver by hepatic lipase, right? Okay, any question? Okay. Yeah, we have more videos, but you guys can just watch yourself. I post it in the Canvas. Okay, I have some questions. All right, so what do you need to know for this lipoprotein session? So major components and functions of the 4 lipoproteins, like chylomicron, VLDL, LDL, and HDL, right? What are the core lipids, which lipoproteins present in each lipoprotein, right? And metabolic fate of a lipoprotein, so you able to understand. We have several diagram, right? Slides, so you can and go through all this diagram and see if you understand how each lipoprotein is metabolized. Basically, how they function to carry or transport a different type of lipids. And what is the role of the LDL receptor This is also important, right? What is the role of LDL receptor? If some people do not have enough LDL receptor on the surface, what will happen to the LDL concentration or levels in the circulation? Will increase, right? So again, I mentioned the receptor number is a key that control the levels of LDL cholesterol, right? And also factors that involve the reverse cholesterol transport process that I just described, right? The ACAT, what ACAT does. What is the function of ACAT? Yeah, esterified free cholesterol, right? To cholesterol esters. And what is this reaction? What is this enzyme? Where in the cells, right? And LCAT in the plasma, right? Carry the same reactions. And CEPT in the plasma, and what CEPT does? Yeah, thank you. So exchange, like lipids, cholesterol ester and triglyceride between HDL and VLDL, LDL. What about the ABC1? The peripheral tissue, right? On the surface of peripheral tissue, and it facilitate transport, right? The lipids from peripheral tissue to the beta HDL. Okay, so SRB1 is also receptor, right? Located in the liver and some other cells as well, surface, and it's help the uptake, right? The alpha HDL. And also, hope you'll be able to distinguish the functions of LPL and hepatic lipase and HSL. They all the enzyme catalyze the same reaction, hydrolyze triglyceride and release fatty acids, glycerol, but they have a different cellular location, tissue location, and this is the main difference, I think, right? Like LPL, basically located in both tissue, adipose tissue and muscle, right? And hepatic lipase, by the name, you know that it's specifically located in the liver. And HSL is also primarily located in the adipose tissue, but also muscle, right? Cell. Some cells that with the triglyceride stored. The HSL basically hydrolyze the triglyceride inside cells, right? And LPL hydrolyzed triglyceride as like in the, the kind of circulation, right? Basically the triglyceride within the two important lipoprotein, right? Which lipoprotein that, triglyceride lipoprotein is hydrolyzed by LPL. Which two, chylomicron and VLDL, right? So triglyceride in both chylomicron and VLDL is hydrolyzed by LPL, right? And HSL hydrolyzed triglyceride inside of that cell, right? So it's different location. A break? Okay. Okay, everybody, let's move on. Okay, I have more questions for you guys. I think this is a new slide. I'm going to post it, right? Don't worry. So hopefully help you to understand, to remember something, right? Which lipoprotein is synthesized in small intestine? Good. Yes, chylomicron. Which ApoB protein is synthesized in small intestine? B48. Yes. Okay. So which lipoprotein is synthesized in the liver? VLDL. And which apoprotein synthesized in the liver? B100, right? Okay, so which apoprotein is required for LPL activity? ApoC2, right? Okay, and which apoprotein is required for binding of LD receptor? LDL receptor? E, right? And also B100, right? Both E and B100. Okay, so just